Hi, today I am going to present in biology the Mendel's law of inheritance or principles of inheritance and variations. So, Mendel's law of inheritance, which is quite popular, and most of the students, even in the school level, they are familiar about it. So, what happens in Mendel's genetics and variation? All living organisms reproduce. It results in the formation of kind. The resulting offspring most often do not totally resemble the parents. Siblings sometimes look so similar to each other, so sometimes even so different. Now, if you want to learn the basic concepts like genetics, inheritance, hereditary variation, it will be worth. So, genetics is the branch of biology which deals with the inheritance as well as the variation of characters from parents to offspring. Inheritance is the process by which characters are passed on from parents to progeny, and it is the basis of heredity. Heredity is the transmission of characters from parents to their offsprings, and variation is the degree by which progeny differ from their parents. Environmental variations are also present. These are acquired and non-heritable. Hereditary variations, these are genetical and inheritable. Now it comes to the about Mendel. He is known as the father of genetics and conducted the hybridization experiments on garden peas for 7 years. And also, he proposed the laws of inheritance in living organisms. During Mendel's investigations into inheritance patterns, it was for the first time that a statistical analysis and mathematical logic were applied to problems in biology. Mendel investigated the characters in the garden pea plant that were manifested as two opposing traits. For example, tall or dwarf plants, yellow or green seeds. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow green seeds, inflated full or constricted green yellow pods, and tall or dwarf plants. The seven pairs of contrasting traits in the pea plants studied by Mendel are shown here also, and why he selected the pea plants. Pure variety where available pea plants are easy to cultivate. Life cycle of plants are only few months so that results can got early. Contrasting traits are observed. Flowers are bisexual and normally self-pollinated. Flowers can be cross-pollinated only manually. Hybrids are fertile. Now, the important terms as you have seen about genetics, inheritance, variation, and then characters. For example, a heritable feature among the parents and offspring. For example, eye color, right? Variants of a character. For example, brown eye or blue eye. Alleles or allelomorph. Alternative forms of a gene. Homozygous and heterozygous concepts. The condition in which chromosome pair carries similar allele of a gene, for example, capital T or small t's. Y capital Y's or small y's. Heterozygous is capital T and small t, capital Y or and small y. Then it comes to the dominant character, the character which is expressed in heterozygous condition. It is indicated with capital letter. Now, recessive character, the character which is suppressed in heterozygous condition and indicated by small letter, as you can see here. Then it comes to phenotype, physical expression of a character, that is phenotype, genotype, genetic constitution of a character, and then hybrid, an individual produced by mating of genetically unlike parents. Now you can also see some more details, and you need to remember all these things. For example, back cross and test cross. What is back cross? The cross between F1 hybrid and one of its parents. Then test cross, the cross between hybrid and its homozygous recessive parent. It is used to identify the genotype of hybrid. Now you can see the laws of inheritance. For example, Mendel conducted some hybridization experiments on garden peas or Pisum sativum, that is botanical name or biological name. Steps, for example, involved in making a cross in peas, selection of two pea plants with contrasting characters, then emasculation, removal of anthers of one plant to avoid self-pollination. This is female parent. Pollination, collection of pollen grains from the male parent and transferring to the female parent. Collection and germination of seeds to produce offspring. Then it comes to the characters. For example, the seven true breeding varieties selected by Mendel's were stem height, flower position, flower color, pod color, seed shape, and seed color. Then contrasting traits were dominant and recessive, tall, for example, dwarf, tall and dwarf, axial and terminal, violet and white, inflated and constricted, green and yellow, red, round and wrinkled, yellow and green. Now, if you want to understand the Mendel's laws of inheritance, then you need to understand the some basics in your family or in your population. For example, Mendel says that genetic inheritance boils down to three simple concepts put forth by Gregor Mendel, a humble monk and part-time scientist who founded the entire discipline of genetics which we see even in modern genetics. Law of segregation, then law of dominance, then law of independent assortment. These are the Mendel's laws of inheritance. Now, here in the segregation, in diploid organisms, chromosomes pairs are separated into individual gametes to transmit genetic information to the new offspring. Dominance, law of dominance, a dominant allele completely masks the effects of a recessive allele, and a dominant allele produces the same phenotype in the heterozygotes and in homozygotes. Now, law of independent assortment, here you need to understand the alleles on different chromosomes are distributed randomly to individual gametes. Now, if you want to understand the inheritance of one gene can be explained by monohybrid cross, the cross between two parents. The cross between two parents' offspring in one pair of contrasting character is called monohybrid cross. Now, crossed tall and dwarf pea plants collected seeds and grew to generate first hybrid generation or first filial generation, that is F1. First hybrid generation or first filial generate plants, F1 plants, tall and non-bare dwarf. For other traits also, F1 generation resembled only one parent and trait of other parent were not shown. Self-pollination in F1 for F1 filial 2 generation to F2 or F2. F2 generation 1/4 were dwarf and 3/4 were tall, identical to parents. F1 generation, one parent trait shown, and F2, both parent traits shown in the ratio of 3 is to 1, and no blending were seen. Now, this is the same thing. It has been shown here in this figure, which you can see here. So, this is representing the inheritance of one gene. Now, you can also understand a cross involving two plants differing one character pair, that is monohybrid cross. And for example, Mendel crossed tall and dwarf pea plants to study the inheritance of one gene. As you can see, tall and dwarf, yellow and green, so cross between tall plant and dwarf plant, then cross between yellow and green plants. Now you can see in different way and what is F1 or first filial generation and F2 second filial generation. These things you need to understand. Now you can also understand the Mendel's what he proposed something is stable being passed to the next generation through gametes, factors, that is genes. Genes or factors are the unit of inheritance, contain the information required to express particular trait. So these are the questions which will be very easy if you understand it for multiple choice questions. Then genes which code for pair of contrasting traits are alleles. Then alphabetical symbols were used, for example, T for tall and small t for dwarf. Plants pair of alleles For height, T, capital TT, and capital T and small t, then small tt. Mendel proposed two breeding tall or dwarf plant, identical or homozygous alleles pair of TT. That is called genotype, which we have already mentioned. Descriptive terms tall or dwarf are phenotypes. Phenotype. Mendel found phenotype of heterozygote our first filial generation was same as parent with TT and proposed in a pair of dissimilar factors. One dominates the other and hence called dominant or recessive factors. Now, another thing it comes are called Punnett square. This is a kind of box which helps you to understand very easily in basic combinations. Production of gametes and formation of zygotes, developed by British scientist Ra- Reginald C. Punnett. Graphical representation. It is graphical representation used to calculate the probability of possible genotypes in genetic cross. Gametes on two sides, top row and top columns. Here you can see phenotype ratio tall versus dwarf 3 is to 1, genotype ratio capital TT versus capital T and small t, then versus small tt, and that is 1 is to 2 is to 1. Now it comes. What is monohybrid cross? Here you can see this represents the self-pollination here is basically representing the monohybrid cross. Then it comes to the test cross. Test cross is the cross between an organism with unknown genotype and a recessive parent. It is used to determine whether an individual is homozygous or heterozygous for a trap. And this is here, you can see it easily. Now, Mendel's law of inheritance. Here you also can understand backcross and test cross. Backcross cross between a hybrid and it's any one of the parent. Then test cross of an organism with dominant phenotype to a recessive individual. And this is here clearly shown here how it looks like. Monohybrid test cross ratio here it shows 1 is to 1, and back cross and test cross here you can see test cross is used to find out the unknown genotype of a character. Here you can see then Mendel conducted test cross to determine the second filial generation. Now, what how if you want to understand further the inheritance of one gene as Mendel mentioned in the laws of inheritance here. Mendel made similar observation. Mendel made similar observation where you can easily understand the concept that how Mendel made similar observation for other pairs of traits where he proposed that some factors were inherited from parent to offspring. Factors are now called as genes. So here you can see F1 or first filial generation, that is capital T and small t when self-pollinated produces gametes T, capital T and small t in equal proportion. During fertilization, pollen grains of T have 50% chance to pollinate eggs of T, capital T and small t. Also, pollen grains genotype T have a 50% chance to pollinate eggs of capital T and small t. And this you can see it easily in Punnett square law. Now, here you can see it again how the ratio can be determined. So Mendel self-pollinated the F2 plants. He found that dwarf F2 plants continue to generate dwarf plants in F3 and F4 generation. He conducted that genotype of dwarf was homozygous and that is a small t. Now it comes to law of dominance. Characters are controlled by discrete units called factors, that is gene. Factors occurs in pair. In a similar pair of factors, one member of the pair dominates the other, that is recessive. The one member which dominates, that is dominant. It is used to explain the expression of only one of the parental characters in monohybrid cross, that is F1, and expression of both in F2. Or it also explains the proportions in 3 is to 1 in F2. So characters are controlled. Now here by discrete units called factors. The factors occur in pairs, and in a dissimilar pair of factors, one member of the pair dominates, that is the dominant, and the second is recessive, that is dominated here or suppressed here. Now, third law, that is law of segregation. It states that when a pair of the law of segregation, it states that when a pair of factors or a character brought together in hybrid, they segregate or separate during the formation of gametes. Alleles do not blend, and both characters recovered in F2 and one in F1. Factors which is present in parent segregate, and gametes receives only one of the two factors. So homozygous parent one kind of gametes, and heterozygous two kind of gametes, each one have one allele with equal proportions. Now, you can also see during gamete formation, the factors or alleles of a character pair present in parents segregate from each other such that a gamete receives only one of the two factors. Homozygous parents produces similar gametes. Heterozygous parents produces two kinds of gametes, each having one allele with equal proportion. So it is a cross. Like in dihybrid cross, it is a cross between two parents offering differing in two pairs of contrasting characters. Cross between pea plants and with round shaped and yellow colored seeds and wrinkled shaped and green colored seeds. That is dihybrid cross. Now here you can see how in dihybrid cross the gametes are formed. So the factors, the similar factor pair of factors or dissimilar pair of factors are present. Now you can also see in details these things, how the inheritance of two genes are possible, and what could be how the Punnett square box will look like. So they are in clear details. So these are the ratios which can be easily derived as a combination series of 3 yellow is to 1 green with 3 round is to 1 wrinkle. So you can also easily calculate these dihybrid cross or inheritance of two genes. Now, what is incomplete dominance? Corens discovered incomplete dominance in Mirabilis jalapa. It is also called partial dominance or semi-dominance. The inheritance in which allele for a specific character is not completely dominant over other allele is called incomplete dominance. The snapdragon or Anti-renum species cross between true breed red flower and white flower F1 generation, pink, and after self-pollination in F2 generation, you can see how in which ratio they appear. So genotypic ratio, same as Mendelian cross, or phenotypic ratio different than Mendelian cross. Now, this is basically the incomplete dominance, where you can understand basically the problem in expression or proper expression. Now you can go for third law. So Mendel's law of independent assortment. So this third law, or the independence of characters, postulates that alleles of different genes segregate independently during gamete formation. This means that the inheritance of one trait does not affect the inheritance of the others. So, this law provides a crucial understanding of how multiple traits are inherited and combined in offspring, contributing to the understanding of genetic variability and population genetics. It is based on, so if you want to understand the law of independent assortment, you can understand here that is based on the results of dihybrid crosses. Here it states that when more than one pair of characters are involved in a cross, factor pairs independently segregate from the other pair of characters. Now, the concept of dominance, you can also understand every gene contains the information to express a particular trait. In heterozygotes, there are two types of alleles. For example, unmodified allele and modified allele. Unmodified allele is generally dominant and represents original phenotype. Modified alleles are generally recessive, and these two eyes are representing the unmodified and modified examples of the alleles. So, if you want further to understand, so consider a gene that contains the information for producing an enzyme. Normal allele of that gene produces a normal enzyme. Modified allele is responsible for the production of something different. So, how you can understand? Normal or less efficient enzyme, in this case, modified allele will produce the same phenotype like unmodified allele. Thus, modified allele is equivalent to unmodified allele and non-functional enzymes or no enzyme at all. In these cases, the phenotype will be dependent. So only on the functioning of unmodified allele. Thus, the modified allele becomes recessive. Now, if you want to understand the examples of incomplete dominance, you can also see these colors of the flowers, how they look like. So these are one of the finest example of incomplete dominance. And here, this is another example. So now you can go for some Punnett square box in case of incomplete dominance, and you can easily understand it. Now it comes to another term that is called codominance. That means they, so from the term you can say they express equally. So both the alleles for a characters are dominant. So and express its full character is called codominance. For example, in blood group crossing, you can see AB blood group of human being and blood group in humans are controlled by three alleles of a gene I. They are IIB and small I. The ABO locus is located on chromosome 9. IA is responsible for the production of antigen A and IB is responsible for the production of antigen B. I does not produce any antigen. So IA and I are co-dominant and dominant over I, as you can see easily here. Now, the details of the blood group crossing, you can see the possible genetic diagram for the inheritance of blood group. This is the best representation which you can see, and these are the combinations, genotypic combinations, which will help you to understand it. So now we can conclude these blood group cross combinations. ABO blood grouping, multiple alleles are present. Three alleles govern same characters. Multiple allele is found when population studies are made. Single gene product may produce more than one effect. For example, starch synthesis in pea seeds controlled by a gene having two alleles B and a small b. Starch synthesis effective if homozygote BB capital B and produce large starch grains, and homozygotes with small b's lesser efficiency in starch synthesis and seeds are wrinkled. Heterozygote with one capital B and one small b, round seeds and intermediate size. Now, thank you very much to helping us to reach more than 1000 subscribers, and I hope that you people will further participate and learn more and more from my channel and visit frequently. Thank you very much.